Today, I will present the first preclinical data from our HLA-independent TCR receptor, or HIT, platform. There are several types of cell therapy binding modalities. T-cell receptors, or TCRs, bind to antigenic peptide fragments presented in the clef of a HLA complex on the surface of target cells. CAR T-cells bind to cell surface antigens using a high-affinity antibody. Because CAR T-cells don't use the TCR, they require artificial signaling domains. TRuCs aim to overcome some of the limitations of CARs, such as the need for high antigen concentrations and tonic signaling, by utilizing antibodies fused to parts of the TCR CD3 complex. Today, I will present data from our HLA-independent TCR, or HIT candidate, targeting mesothelion. This is a fully human TCR that binds a cell surface antigen and activates the T cell for killing. Mesothelion is a 40 kilodalton cell surface protein expressed in mesothelioma, pancreatic, ovarian, and other cancers with limited expression in normal tissue. We used our proprietary naive phage display libraries expressing fully human alpha-beta TCRs and biopanned these against recombinant mesothelion. We identified 4 TCRs that were highly converged and produced these in E. coli. One TCR bound to mesothelion is measured by surface plasmon resonance with a KD of approximately 1 micromolar. Whilst this affinity is in the range of an optimal TCR, it is still much lower than that of antibodies, which have low nanomolar to picomolar affinities. Next, we determined if our HIT could elicit activation in a T cell. We transduced our HIT into Jurkat T cells and measured mesothelion-dependent activation. No activation was detected with our K562 control cells. Activation was detected with our mesothelion-positive HCT116 cells, and significant activation was detected when K562 cells were transfected with a mesothelion-expressing vector. These are experiments from a single healthy donor, whose T cells were transduced with our candidate HIT. We co-cultured these with a panel of antigen-presenting cells with varying levels of mesothelion transcripts. The HIT T cells produced both interferon gamma and granzyme B at levels that correlated with mesothelion. We have since been able to affinity optimize this HIT to alter the response to cell lines depending on the level of mesothelion expression on the cell surface. This is a key step towards ensuring no on-target, off-tumor reactivity. We then co-cultured our HIT T cells with mesothelion target cells in a killing assay. Compared to the control T cells, the HIT T cells kill both the antigen-positive CAPAN2 cell line as well as lung PDX model with maximal killing at around 24 hours and 48 hours. Naturally occurring TCRs rely on the interaction between CD8 and the HLA complex to elicit function, and when CD8 is blocked, there is little or no response to antigen, and CD4 cells don't kill at all. Our HIT T cells don't need CD8, and both CD8 and CD4 T cells are activated in response to mesothelion target cells. We also compared our HIT with an anti-mesothelion TRuC T cell, made in-house and transduced into T cells using our lentiviral vector and standard processes. These cells also function independently of CD8. CAR T and similar therapies, which rely on high-affinity antibodies, have been shown to be neutralized by soluble target protein. The assay on the right demonstrates that our TRuC T cells are inhibited by increasing levels of free mesothelion, but our HIT is not neutralized. This is an important feature because many cancer-associated cell surface proteins such as mesothelion are both membrane-bound and secreted. Lastly, we sought to determine if our HIT T cells will be efficacious in vivo. Immunodeficient NSG mice were implanted with human pancreatic CAPAN2 mesothelion-expressing tumor cells, and T cells were administered by a single IV injection. Mice were treated with our HIT T cells with increasing doses or injected with control cells. Strong, dose-dependent and persistent tumor regression was evident at doses greater than 1 million HIT T cells, as shown in green. We also tested our version of the anti-mesothelion TRuC T cell, shown in red. Our HIT T cells demonstrated superior efficacy to the TRuC even at lower doses. We've demonstrated that we can identify and engineer HLA-independent TCRs, which gives us access to CAR and TRuC targets. The lack of inhibition by soluble protein, the fact that HITs use natural T-cell binding and signaling, our ability to engineer and optimize TCR affinity for safety and potency, leads us to believe that HIT T cells are an exciting new therapeutic modality that could be transformative for people with cancer. These proof of concept experiments will form the foundation for preclinical testing for future HIT development. I would like to thank all of my colleagues at Adaptimmune and Astellas for their contributions to this study.